CMU, and I'll be talking about high-throughput framework to detect synapses in electron microscopy images.
Thank you. Hi. Thanks for being here. I wanted to tell you a story today about a computational and experimental problem that we came across recently while trying to study changes in synaptic development in the brain. So just for some quick background, so synapses are connections between nerve cells or neurons in the brain, and they allow cells to communicate with one another and to transfer information to one another. So in our brains, just to give you an idea of the scale, we have about 100 trillion synapses in our brains, amongst about 100 billion neurons or so a massive amount. And synapses are important because changes in their properties, and in particular their density or their distribution in a region, and in their individual strength, is thought to underlie learning and memory in the brain and can vary according to ages, according to different diseases, or sensory experience. So just as an example, you can see on the bottom right here, bottom left, a very well-known process that happens throughout many brain regions, where during development you see a rapid explosion in the number of synapses in this particular brain region, followed by a period in which 50 to 60% of the synapses are pruned away, they're thrown away. And so you reach some baseline level by adolescence, but if you see insufficient pruning, people have shown that this is one symptomatic factor of autism spectrum disorders. And on the flip side, if you see too much pruning, this might be indicative of another neurodevelopmental disease called Rett syndrome. Synapses are also very plastic, so they can change their efficacy based on sensory experience. Or for example, in mice, people do a lot of experiments where they deprive a mouse of a particular whisker, and they look how changes in synaptic organization follow that. Drug treatments, these are all common problems that neuroscientists are looking at today, and it's kind of driving the study of the brain today. So what are the experimental techniques that people are using to detect or count synapses in the brain? So I'm going to plot them here for you on two axes, one showing how fast we can collect the data, and the other showing how fast we can process or analyze that data. So one of the classical techniques, electrophysiology, is where you take individual electrodes, you puncture cells, and then you test whether or not a connection exists between them. So this is a great technique. You can definitely detect synapses and a lot of other properties of synapses and neurons. But the problem is that you can probably do this on maybe 5 to 10 pairs of cells per day. And if you're lucky, you might find 1 or 2 synapses. So in terms of the data collection, it's a very slow, laborious process. The other extreme is to just do imaging. So you can take thousands and thousands of EM images, electron microscopy images, and you can actually see in the entire ultrastructure of what's going on in the brain. So the challenge here has mostly been in the analysis, because you're getting every preservation of every single structure, and you're looking for these very small signals. So this is what a synapse looks like in an EM image. And up till now, there haven't been fully automated ways to detect these small bands of postsynaptic densities in these very noisy and heterogeneous EM images. So it has really hampered our ability to do this on a very high throughput way. In the middle here is MRI-based approaches, which are just simply too coarse to actually detect synapses. Recently, there have been a few other technologies proposed for us. So the MGRAS technique was talked about here last year at ISMB, but it requires a specialized transgenic line that stains or highlights particular synapses in fluorescence images. Array tomography is also a new technique that actually allows you to differentiate between many different synapse types, but it's also struggling with this analysis technique, and it's a cumbersome process because you're doing sequential immunohistochemistry across many different antibodies, and it's just a tough process to do. So we just kind of asked ourselves, well, it would be nice to be up here, and what I mean by up here is we have 2 criteria. We want to be able to count or detect synapses. We want to be able to measure their strength because of the applications that I mentioned earlier. We want to be able to collect the data and analyze it quickly. And if there's one place we're willing to sacrifice, it's going to be, okay, maybe I can't differentiate between all different, 20 different synapse types, or maybe I can't detect the failure rate of the synapse, or maybe I can't resolve a synapse to an exact pre- and postsynaptic neuron. But that's okay, because there's still a lot of stuff I can do with just this data. So one day we were just kind of browsing through the imaging literature, and we found this old paper by Floyd Bloom and George Agajanian, published in Science in 1966, almost 60 years ago. And what these guys did was, so here I'm showing a zoom in of what a synapse looks like in a conventional EM image. So you can see the dark postsynaptic band and the vesicles on the presynaptic side. And they treated these images with something called ethanolic phosphotungstic acid. And so what this stain does is it targets proteins, which I'm showing here, that are prominently on the pre and postsynaptic side of the synapse. And ethanol is used to basically wash away everything else that's in the image. So you see this much cleaner image where the synapse is preserved and all the other stuff is removed. Okay, it's not that simple. So we spent several months in the lab trying to reproduce this assay. And we did pretty good at it. So here again is our conventional EM image where it's hard to see the synapse. And here are two samples of using this EPTA-based EM technique where you can see the synapses here are much easier to identify here compared to here. But you are seeing still a lot of preservation of other structures. So here's a nucleus, for example. I mean, proteins are everywhere in the brain, so it's not like you're just getting the synapses, but our claim now is that being able to do automated processing on this right-hand side, going from this step to this step, is going to be much easier compared to what you see using conventional EM. Okay, so I'm going to talk about now how we go from these original raw images to these automatically classified images. So the main challenge in doing this is that despite the fact that you're specifically staining the synapses, there's a lot of variation across samples. And this variation could be just due to biological variation in the tissue, in the age, or the particular brain region you're looking at. It could be in variation in the actual chemical reactions of this technique. It could be through variation through the image acquisition process, so the type of microscope you use, the illumination, the focus. And so I'm going to just walk you through 4 kind of standard steps in our classification pipeline on how we go from these original images to the classified step. So the first thing we do is a very conservative image segmentation step. And the idea is that, look, you know, we're preferentially staining these synapses. Let's get rid of 90% of the image and just focus on the dark spots. And so instead of trying to manually pick thresholds on what to throw away and what to keep, we do an intermediate step of an adaptive histogram equalization. And what that does, it enhances the contrast in each local window slid across the image to match a flattened histogram. And then these windows are combined using bilinear interpolation to smooth the boundaries between these windows. And so what's nice about this technique is that if you have some artifact that's maybe dominant in one region of the image, it's not going to mess up your histogram over the entire image. It's just going to be localized. So this allows us to now do a binarization of the image using a single sample independent threshold to generate what we're calling candidate synapses or candidate connected components or blobs. And so just to note that in this step, we lose basically maybe 1% of the synapses in the original step. And this is almost always because you have 2 adjacent synapses that are merged into the same connected component. So we're fine with that. Okay, so the next step is, so here we have these candidates. And here I've highlighted a bunch of candidates across a few samples. And this is just showing the sort of heterogeneity we're dealing with. So these are all synapses and these are non-synapses. And you can see the variation in the staining here. You can see it's much more illuminated. And before we built our classification framework, we wanted to try and reduce some of this heterogeneity. And so we did 3 things with that. So first we extracted a window surrounding the synapse, a 75 by 75 pixel window, to give you some contextual information about where this synapse lies. Because there were a lot of dark spots, for example, in the nucleus, but that has a very different context than what these synapses have. So this is it's a key indicator of being able to differentiate between the 2. Then we do a standard normalization technique, and then because these synapses have an orientation, we apply the hue transform to get all the synapses aligned along the same edge gradient. So we do this process, and you can see that how much we're able to reduce the heterogeneity. So this is a much more homogeneous set of examples that are going to be much more amenable to classification techniques. Okay, so just quickly, I'll talk about the features that we're using in this classification step. And they fall into two different classes. One being intensity-based features. So for those, we use texture, which is a very common cue that people use when doing manual annotation of these EM images. And we basically just use the MR8 filter bank proposed by Varma and Zisserman in 2004. And then we use the very common histogram of oriented gradients that finds edge directions and the magnitude along those, proposed by Dalal and Triggs in 2005. Okay, so these are our intensity-based, but what's also interesting is that if you just look at these blobs and the shape of these blobs, those can also be an informative indicator of whether or not this might be a synapse. And what I mean by that is that synapses typically have this long elongated shape to them. So you can extract features, for example, the length, the width, the perimeter, the area, and others. You can look at the rest of these features in the paper. But we have 10 different features characterizing what the shape of this blob looks like. So we have two different, the intensity-based features and the shape-based features that are independent. And overall, each one of these candidate windows is represented by this 480-dimensional feature vector that we normalize in RN. Okay, so the next step then is to just build a classifier. So we have our feature matrix for all the synapses that we found. For the non-synapses, we feed it into four different classifiers. We use an SVM, a random forest, a boosting technique, and then we also compare against a template matching technique that is often used in these classification problems. Okay, so we have this technique, we have this pipeline, and we wanted to test it on some representative region in the brain that a lot of people care about and might want to study. So we chose this very interesting region, the somatosensory or the whisker cortex in the mouse. And what's really fascinating about this region is that each one of the whiskers in the mouse is somatotopically mapped in a one-to-one fashion to a column of neurons in the somatosensory cortex in the brain. So here you can see a schematic of all the whiskers on one side of the mouse face, and these are the individual columns corresponding to each one of these whiskers. So there's an exact mapping here. And the advantage of this is now if you do, so here I'm showing a piece of tissue of this region, and you can exactly identify this same region in old mice and young mice across all these different animals. So you can ensure that you're really capturing the exact same region. I mean, there are some depth issues here, but more or less capturing the exact same region across all these different animals. So it's one way to control for the variability that you would see otherwise. Okay, so we can dissect this. We chose this D1 barrel kind of arbitrarily. We can dissect it out, and then we imaged it using this EPTA EM technique. And so we did a sample of 6 animals, 2 at postnatal age 14, so 14 days after birth, 2 animals at P17, and then 2 at P75, well into adulthood. So for each animal, we took about 130 images covering about 3,000 square microns of space in the brain. Okay, so how well does this pipeline perform? So we gathered some training data. We looked at our two animals at P14, 2 at P17. We counted manually, we labeled 11% of these images, counting about 230 synapses and 2,000 non-synapses. We did 10-fold cross-validation, and on the right here you can see the AUC under the ROC curve and the precision recall curve. So the ROC curve includes benefits if you predict negative examples as negative, which is not as informative. So let's just talk about the precision recall. We see about 74%. And using this default classifier threshold of 0.5, this is for the SVM, we get about an 84% precision and about 67% recall. Okay, so not bad, but people were like, well, how do we know that you chose these true synapses? Maybe you just chose the best ones and made it easy for yourself. So we're like, okay, fine, why don't we get an independent expert to come and label these images, and then we'll run our classifier on those and see how well it performs. And we basically see the exact same, very similar numbers in that. So, okay, so these numbers are not bad, but we started using this technique, and we kind of came to this interesting problem. So what did I just describe? We have some sample. we labeled a very small subset of the images, and we made some models, some classifier using those. And now what's going to happen is somebody's going to come, they're going to use this EPTA technique to generate a new sample B, and this sample might be in a different brain region, a different time. You're going to see a lot of these variations that I discussed before. And the question is, how well is this model going to perform on these set of images? So we did do some things to try and adjust for the heterogeneity that you see, but the ideal thing to do would be basically label some of these images, build a new model, and then use that again. Okay, but we don't want to have to ask users to have to build a model for every time they generate a sample. So what are we going to do? And how do we, so the question we asked is it possible to improve this model by somehow leveraging the fact that I have thousands and thousands of unlabeled images just sitting there? Okay, and so what we did was we turned to semi-supervised learning. And so in particular, I'm going to describe a co-training algorithm proposed by Blum and Mitchell. And they have a lot of interesting theoretical properties of this algorithm that you can read about. But the basic idea is the following. So I have some labeled images from my original sample A, and I have these two different independent feature sets. So one based on the intensity features and one based on the shape features. So instead of building one single model with all these three different feature types, I'm going to split it into two and build one model for each one of these feature types. And then I'm going to apply each model to the unlabeled images from the new sample that I have. And what is this going to produce? So for each one of these candidate windows that I have now, this model is going to give me a confidence value of how likely it thinks that this guy is a synapse. Okay, and so what co-training says to do is, look, if I have two models and both of them are super confident that this guy is a synapse, let's just assume that it's right and add this into our set of positive examples of synapses. And likewise, if both models think that this is not a synapse, then we're going to add that to our set of negative examples and assume that it's correct. And we're going to discard the stuff in between. So now what do I have? I have some labeled images from sample A that I started with. I have my co-trained set of positive examples, my co-trained set of negative examples, and I'm going to retrain a single model using all these three different sets. Okay, so does this help us? So what I'm going to show, what I show here is here's building a model using our P75 animals, testing it on the P14 animals, and this first row is no co-training, so just carrying the exact same model over, and we get some performance. The next two lines show how co-training improves the classification accuracy, and this percentage I'm showing here is you have to define some threshold of how many images that you take from this set and this set to include in your new classifier. And that's what's changing. So 1.5% means more and 0.5 means less. And the take-home message here, and this is the same table but just flipped around when I train and what I test, that this co-training process increases the accuracy on detecting these positive examples by about 8 to 12%, and the AUC goes up also by 1 to 4%. But if you impute too many examples, you can actually see a decrease in performance. So you need to be careful about how you use this co-training in terms of just taking the most confident examples that you see. Okay, so now we have this, and the next natural question was, can we actually discover some interesting new biology using this technique? So we looked at, as I was discussing before, about developmental changes that happen in the brain. And so one of the very fascinating ones is this synaptic pruning, where you see a rapid explosion in the number of synapses followed by a drop-off. And people have quantified this using other techniques very slowly. And so here in a matter of about a week or so, we looked at our three different age of animals, P14, P17, and P75, and I'm plotting here the frequency or the number of synapses across each of these different ages, and you see exactly this big peak that happens at about P75, followed by a drop-off by the time you reach adult at P75. So this is very consistent with other data showing that, describing pruning. I also plotted here the size of the synapse as a proxy of its strength, and how much it would depolarize the postsynaptic cell if fired. And people have also done very laboriously using electrophysiological techniques on the distribution of synapse sizes or strengths that they see with this data. And people have showed over and over again that it follows this log normal distribution. And using our images, just taking the size of this blob as a proxy for strength, we also can replicate very robustly this log normal shape. Okay, so the point here is that synapse density and morphology are important characteristics in a lot of neurobiological problems, and we can infer both of these using our framework. Okay, so just to conclude, we reproduced this 60-year-old EM technique to selectively stain synapses, and we coupled this with high throughput and fully automated analysis. So the advantage here is that we don't need any specialized transgenic animals. You don't need a very cumbersome experimental technique. It's very feasible for large or small labs. All you need is a basic EM facility. We use this technique to study changes in synapse density and strength in the developing cortex, and we believe that it might be able to be used to look at drug-related changes or other plasticity-related changes in the developing brain. We use a very standard classification, normalize, extract features, classify. But we also think that this semi-supervised idea is a good one to generate robust classifiers, especially when you have tons and tons of unlabeled data, as we typically do in bioimaging problems. Just some acknowledgements of my co-authors, Joe, and my advisors, Alison Barth and Ziv Bar-Yosef at CMU. Thanks to Fernando and Gustavo for image processing advice, and Patrick for doing the manual annotation. And thanks to the NSF for the travel funding that they provided. Thanks.
All right. Questions?
So if you have this 60-year-old technique, why aren't people just using it?
Yeah, so I mean, there were, if you look at the citations of it, there are maybe a handful of people who are still using it, but I think it was just long forgotten. I actually emailed the authors of the technique basically this exact question. I was wondering, like, is it actually legit, or maybe there's a reason people have stopped using it? And and he told me that his goal was actually to do this automated analysis. He was at the NIH, I think this was in 1966, and he said he just didn't have the computing power at the time, so he moved on. But we just sort of found it and we went with it. So.
One question, one correction. 66 is 47 years, not 60, but anyway. So that's.
Oh yeah, good point.
But the question is, by analyzing the parameters of the extension of the and the parameter that you have for the synapses, can you deduce some characteristic of the synapses, or if you try to extend the analysis just to see if you can see different kinds of synapses, shapes, and their synapses? I mean, you know that there are different morphology of synapses in there. Is it possible with your technique to approach this kind of thematic?
Yes. So one thing that we found is that some synapses have this very prominent presynaptic density. So you see like two bands of synapses, which are characteristic of symmetric synapses or inhibitory synapses. But we're not sure. I mean, this was also a question that I asked the original authors, and they suggested doing some immuno-EM where you can actually label against some GABA particles, where you can label them and actually see what inhibitory synapses look like using this technique. I mean, we know a lot about what they look like using the standard EM, but there hasn't been as much work looking at.
Okay, we try to stratify, for example, for the length and the width of the signal. So to see that if you're stratifying the data by the parameters that you have for the analysis, you will know classes.
Yeah, we see three different classes. I don't have a slide for it now, but I guess maybe I could just point out. So we see some synapses that look like this, where you see this prominent postsynaptic density and these little blobs, these little dots to the left of it that we think are vesicle release sites. Then we also see these where you just see the band itself, and then something like these where you see a very dark band and a lighter band on the presynaptic side. So we think these are three different classes of neurons. We don't know what they correspond to biologically, but we're trying to investigate into that now.
More questions? So in between, I have one question. Whenever you have one very clearly defined class and one that is not so clearly defined, one option would be one class classification.
Mm-hmm.
Did you consider that? One class like one class SVMs or one class classification in general?
Like in terms of the synapses and the non-synapses, or?
Would it be an option, or?
Sorry, I didn't
Okay,
follow.
so it's not a big deal.
Okay.
I was still wondering a couple of things about your SVMs, random forest data boost. I mean, all of these algorithms have parameters. Do they have the same level of optimization?
So yes, so it's true. I mean, when I did the SVM, I did and I didn't talk about it, but an extensive grid search on trying to optimize the parameters. The random forest also had the same, but I did just standard grid search for that, over tenfold cross validation.
I was also wondering, co-training showed huge improvement there. Did you ever double check on like an independent test set or something? Not just cross validation, but some left out.
So the ones that we tested on were the manually labeled ones. So they was left out. Yes.
Okay. Because I mean, sometimes you have to be a little bit cautious. There are positive. I mean, there is experimental results, for instance, where cross-validation together with stacking produced like non-significant improvements.
Yes.
And for some learning settings, you really have to be very cautious.
Mm-hmm.
Questions? More questions? There's one.
Actually, just have a question about, you said you analyzed a few of them and answered them yourself, and then this expert came in. Did you ever check agreement just to see, in that sense, like how well a human does those tests?
Oh, so like.
Presumably can't be.
Oh, I see.
Because I know the previous talk, and I mean, I had the same experience, sometimes.
Yeah. So you basically take two humans on the same data set and. Yeah, but I don't. So we didn't do it on the same image. We we did it at per image just to generate the training data. But yeah, I didn't try that.
Okay, maybe last question.
My question is on the part of classification. Since I would be interested to know if you thought to use a supervised approach, since you have some data that are your test set for good embed samples, you could embed these in one dimension. And I imagine that they are ordered in one dimension if you have a good embedding. And then you could see where your synapses are falling down on that dimension. So a completely unsupervised approach where you embed one dimension and you see the anonymous synapses where they are falling down in dimension.
So that was my original attempt. I mean, I thought, I didn't want to do all this labeling myself, so let's try and do something unsupervised. And I found some sets of parameters that work pretty well, but then the main problem was that these parameters didn't generalize when I got the next sample, and I saw a lot of variability in the stain. So I mean, in theory, yeah, there's some way of embedding this into another space where that's going to be preserved, but we had a hard time doing it without generating at least a little bit of training data.
What did you use to do this one as when you tried to do this kind of evaluation? Which kind of algorithm?
So we also tried a segmentation. So like similar to what we started here, we do kind of a first pass segmentation to generate these candidates and then look at different properties of each of these candidates to see, what's the most which are most likely synapse lookalikes. And I mean, the template matching that we compared against is also very much like that, where you have some templates and for each candidate you're going to see, does it correspond or does it look like any of these other templates, and it just wasn't as robust as we thought.
I was saying something as performing embedding in one dimension.
Maybe we can take this offline.
Okay, we can talk.
Well, I think everybody's looking forward to having a coffee now. I would like to thank all four speakers again for this great session.